Well, class, chapter 17 is about how you get from genes, these are genes, to protein molecules, like protein, my glasses or the mouse. So these are protein molecules. You get it from the genes. You remember last time I had the ladder, showed it to you. That was the gene, the DNA is the gene. We talked about this, and then you get the protein molecules. So let's move on. So the flow of energetic information, the information content of genetic, we talked about this. The DNA inherited by an organism leads to a specific trait dictating the synthesis of protein molecules. Okay, we talked about that last time. Proteins are linked between the genotype and phenotype. Right. So if my color of my eye is blue, then that is a phenotype. Blue is a phenotype, but the genes said it that this has to be blue. Remember we talked about that. And then this, my color of my eye, which is blue, is a protein molecule that make it blue. It's a protein molecule that makes it brown. It's a protein molecule that makes it green. Okay, the variety of color of eye we have. So the protein molecules, my skin is protein molecule, okay? And they dictate what gene is supposed to be expressed, okay? Here is a gene expression, the process by which the DNA directs protein synthesis, okay, includes two stages, transcription and translation. So as you can see, the albino deer is different than the normal deer, brown deer, because of the protein molecules making this deer albino white, I should say white. And then the protein molecules on this deer are making him brown. I hope I'm making some sense. Same thing here, an albino raccoon, and so on and so forth. The basic principle of transcription and translation, RNA is bridge between gene and the protein found, you will see it. Transcription, the definition of transcription is RNA synthesis. That's transcription. And the definition of translation, it is protein synthesis. Protein synthesis versus RNA synthesis. You should know the difference. Messenger RNA, as the name applies, is a messenger that takes the information from DNA and take it to the protein molecule and synthesize. This would be, remember last time I had my pen with me, this would be messenger RNA, this is DNA, messenger RNA, and then you have protein. So this is DNA, RNA is synthesized based on DNA. So that's transcription. And then based on the RNA, protein is going to be synthesized. That's called translation. I hope I'm making some sense. And of course, the site of the protein synthesis is in the ribosomes. Ribosomes, that's where everything comes together, all three, and the amino acids, the three RNAs come together there, and the amino acids come. In prokaryotic, translations of mRNA can begin before transcription has finished, yes. Okay, here's, they call it a central dogma in biology. So here is a DNA, here is transcription, synthesis, making, you don't like the term synthesis, making the RNA based on DNA is called transcription. And then when the RNA goes into the cytoplasm in the ribosome, right here, it goes through the ribosome, the RNA goes through the ribosome, and amino acids are going to be attached to each other. These blue balls are amino acids. When these amino acids are attached to each other, then that process is called, that process right here is called translation. The process here is called transcription, making RNA based on DNA. And that's called central dogma of in biology. And they're explaining it right here, nice and beautiful. This is in eukaryotic cell. This is in prokaryotic cell. They're showing you that this is happening. They do not have, prokaryotes do not have a nucleus like here. So in eukaryotic cells, transcription takes in nucleus and translation takes in cytoplasm. in a prokaryotic place, they both taken in the cytoplasm. A primary transcript is the initial RNA that transcribes from any gene prior to processing. The central dogma is the concept that I talked about it, DNA, RNA, protein, central dogma. Okay, how are the genetic code? How are the instructions of assembling amino acids takes place? Yes, DNA, there are 20 amino acids. Make sure you already know that from exam number 1. In us human, there are 20 amino acids and there are only 4 nucleotides, A to T, G to C, right? All of the living organisms are made up of these 4. Bacteria, whales, sharks, elephants, mammoth, which was on the planet Earth. Now they are not anymore. Okay, 4 nucleotides. Now, the number of amino acids in us 20, but all of the living organisms, I read it somewhere, I wish I've read, it's more than that, it's 100, 120 different amino acids, but how many nucleotides corresponds to an amino acids, we will see that. So right here is a DNA template. And if DNA is A to T, C to G, C to G, A to T, A to T, A to T, C to G, and G to C, T to A, I just don't want you guys think that on one side they're all A's or on one side they're all C's. No, that's one student thought one time before, and I said, no, please don't think that. You know, it's just different, but always A to T and G to C. Doesn't matter which side which. So having said that, if this is the template to make the RNA, since RNA does not have T, it would be U, C, it would be G, C would be G, A, no T, so it's U, A, U, C, G, C, G, G, C, A, U, G, C, and T, A. That's what the mistakes you make during the exam. You think it's a T right here, and you put down U here. Then you get a different amino acid at the end. You will see that. Okay, so these 3 nitrogenous bases on mRNA, UGG, it's a codon, it's called codon. 3 nitrogenous bases on mRNA is called codon. These 3 will give you an amino acid tryptophan. These 3, UUU, it will give you phenylalanine. GGC will give you glycine. UCA, I'm reading from left to right. Remember that? If I read it from right to left, it's going to be a different amino acid. But reading from, and I have to say that during the exam to you, reading from left to right. Reading from left to right is UCA, that is serine. Okay, so this is right here from DNA to RNA is transcription. From RNA to protein molecules, polypeptide right here is translation. I hope that makes sense a little bit. Okay, so Amir, the question is, how did you say, how do you know UGG is tryptophan? How do you know UUU is phenylalanine? How do you know GGC is glycine and UCA is serine? They call it the codon table. The first letter is U, second letter is U, right here, last letter is U, so that's right here, UUU is phenylalanine. Let's go back, first letter U, GG, let's go here, first letter is, first letter is U, second letter is G, right here, and third letter is also G, right here, so that would be tryptophan. Serine, the next, oh, come on. UCA, I hope you remember that. Your first letter is U, C, UCA, right here. These are all serine. UCA, last letter is A, right here. UCA, serine. So you should know, while I'm here, I should mention two other things. One of them, start codon, is AUG. So if I go back here, always AUG, sequences starts with AUG. When the school starts, easy to remember. When the school starts, month of August, AUG. So protein synthesis always starts with AUG, with methionine, right here, methionine. And then at the end, if you go back, at the end, there are 3 codons that is for stop. U, A, U, G, A, U, A, G. So at the end, it has supposed to be UAA or UAGA or UAG. Those are 3 codons for stop. Okay, you should know these things. I don't expect you to memorize this table. Do not, please do not memorize that table. During the exam, if I want to ask a question, this is the DNA. What is, I give you the DNA, and what is the amino acid sequence? I give you the DNA, one strand, one of them, the gene. And then I say, what is the amino acid sequence? I will give you this table too. Okay, so I will give you the RNA and say, what is the amino acid sequence? So these are based on the codon. Later on, you're going to learn anti-codon, and then you're going to get confused, and I hope you don't. So these are the mRNA right here, mRNA, the codon. I hope. Okay, let's move on. Evolution of the genetics. The genetic code is nearly universal, shared by the simplest bacteria to the most complex animals. Already said that genes can be transcribed and translated after being transplanted from one species to another. Yes, so they can, the fruit fly gene, not the fruit fly, the firefly gene can be transported into plants, and the plants glow, or to a pig, and the pig can glow, so on and so forth. So it is possible nowadays to do that. Molecular component of transcription, RNA polymerase make RNA. You remember we talked about RNA before, the primer and the enzymes and so on and so forth. But here we go again. We are revisiting some of them a little bit. Makes RNA polymerase makes RNA does not need primer in this case, because of this is protein synthesis, and then opens up DNA. So DNA is, alpha helix. It has to open up, not the DNA polymerase opens it up like last time. The RNA polymerase comes and open up the DNA. Pair up uracil with adenine. Remember, of course, you know that. And the DNA sequence where RNA polymerase attaches is called the promoter. In bacteria, the sequence signaling the end of the transcription is called the terminator. Right here, so this is the promoter, and we'll talk about in gene regulations. We'll talk about this a little bit more, but right now, let's leave it for that. And then the RNA transcript is right here. And then, of course, this is unbound DNA, and then the RNA, here we go. I was looking for this thing. For RNA polymerase is the name of that here, huge entire protein molecule, which is an enzyme. And as RNA polymerase goes through, you have a promoter, of course, here. It's compound, they call it downstream, the term that I attended a few meetings, they use it a lot, downstream. This is upstream right here. So down it goes, the RNA polymerase goes downstream, and the RNA is made up. And remember, there are 3 codons in here. What is this? And completed transcription, and everything, the DNA goes back to its original shape, and the RNA is synthesized, and the DNA goes back to its original shape. Oh, sorry, I think this is a video. Yes, let's do it.
The first step in protein synthesis is transcription of RNA from DNA. The portion of the DNA that is transcribed into an RNA molecule is called a transcription unit. An enzyme called RNA polymerase carries out transcription. With the help of protein transcription factors, it attaches to the beginning of a region of the DNA called the promoter, pries the DNA strands apart, and untwists a short portion of the double helix. RNA polymerase moves along the DNA, pairing up RNA nucleotides with their DNA complements, adding nucleotides to the end of the growing RNA molecule. Here's a close-up view of elongation. Only one DNA strand, the template strand, serves as a template for RNA synthesis.
Only one.
RNA polymerase moves along the DNA strand in the 3' to 5' direction, adding nucleotides to the 3' end of the RNA chain. Note that U in RNA pairs with A in DNA.
Okay, so this is...
Once transcription is complete, RNA polymerase releases the completed RNA and detaches from the DNA.
Okay, how does... The question is, how does RNA polymerase knows this is the end? Okay, you will see that in next chapter. That's all gene regulations. You will see that in next chapter. How does it start? You saw the promoter, right? Here, there is a segment of sequence of nucleotides, which is called promoter. Okay. So these are the things. And at the end, the RNA polymerase knows when to stop. And that's called the stop signals. You will see it in next chapter. You might see some of it in here too. Here we go. Okay, so you have the promoter, which is called a Tata box. The promoters are Tata box. It means the RNA polymerase sees a Tata box, it goes boom, let's attach to it, and then continue on and do a transcription. So Tata box is what is Tata box? It's a lot of TA, AT, TA, AA, AT, 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 and so on and so forth. There's a lot of them. Here they showed you just a little bit of it. And then this, of course, is a non-template strand of DNA. This strand that is going to get transcribed right here. This is the template strand. This is start point. Again, you will see it, and eukaryotic promoter, several transcription factors to DNA. So these are transcription factors. And then here it is, RNA polymerase 2, and transcription factors are another thing that they said in a video. I did not say it in the previous video. So the transcription, it goes from 5' to 3'. So new RNA are added to the end, the 3' end. Okay, look at the DNA. The DNA that is being transcribed, sorry, what happened? The DNA that is being transcribed, the template that is going to be transcribed is 5'. Same thing as you learn in the DNA duplication. You remember, same thing. It's 5' in here in DNA. It's 5' here is DNA. In RNA has to be 3'. So anyhow, let's go. Elongating of the RNA strands, and RNA polymerase moves along the DNA. It untwists double helix 10 to 20 bases at a time. If you want to know, transcription progress at the rate of 40 nucleotides per second. Remember, for DNA it was 50 in eukaryotic cells. So 40, this one for protein synthesis is 40, a little bit less. So per seconds in eukaryotics, a gene can be transcribed simultaneously by several RNA polymerase. Yes, so that is possible too. Nucleotides are added to the 3' end of the growing RNA, which I already talked about that. Okay, so pretty much here I talked about everything. Make sure you understand and you know the matching. This is the DNA is T, the RNA is A, DNA is A, the RNA is U, the DNA is G, the RNA is C. Make sure you know that. Okay, so termination of transcription. So how does it ends? I talked about that briefly a little bit, but let's see what this PowerPoint have to say about termination. The mechanism of termination are different in bacteria and eukaryotes. In bacteria, the polymerase stops transcription at the end of the terminator, and the mRNA can be translated without further modification. In eukaryote, RNA polymerase 2 transcribes the polyadenylation. Okay, it means at the end, you have many A's. Poly, it means many, as you know. Adenylation, it means adenine. A. The RNA transcribes is released 10 to 35 nucleotide past this polyadenylation sequences. Right. Each end of the mRNA, why you call it pre-mRNA, you will see the mRNA that is synthesized, pieces and bits of it are going to be cut off. They're not needed anymore. That's what they call it, pre-mRNA. So each end of the pre-mRNA molecule is modified in a particular way. The 5' end receives a modified nucleotide 5' cap. You will see a picture, and the 3' end gets a polytail. So you see that these modifications share several functions. They seem to facilitate the export of mRNA to cytoplasm. They protect mRNA from hydrolytic enzymes. And then, of course, in the cytoplasm, that's what happens. And then the ribosomes attached to the 5' end. Right here, so all of the orange right here, I don't want to draw too many lines in here, it can be confusing. All of the orange is the RNA. The whole thing is pre-mRNA. Look at the poly-A tail. So that means this is the end of it. Okay, and then of course the cap, it goes into the cytoplasm with a cap, and you will see that a little bit. So that's a guanine right here, 3 guanine triphosphate. You will see that, and then this is a 5' end. This is a 3' end, and you should know this 3' UTR untranslated regions. So these are not gonna, these regions, polyadenylation occurs here as well. Signals, stop signals is right here. Do you remember UAA? I talked about that, UAG or UGA. They're there. So the regions that includes a protein coding are these regions. This is start signal. And then these are the protein. So UTR stands for untranslated regions. And then this whole entire thing is called pre-mRNA. So the next thing is introns and exons. Split genes and mRNA splicing. So introns, non-coding regions. It means they do not, they stay inside of nucleus. Introns stay in. Exons, they get translated. They go outside of the nucleus. They get translated. Okay, right here. And then RNA splicing removes introns and joins exons. That's what the term RNA splicing means. Segments of that segment and that segment, they get connected together. And that segment goes away. So spliceosomes is a protein molecule which are several small nuclear ribonucleoproteins, SNRNPs they call them, that recognize the splice sites. Ribozymes, so far in this semester, you learn that the enzymes are protein molecules. Now, there's one exception, and that one exception is ribozymes. Ribozymes is the catalytic RNA molecule that function as enzymes and can splice the RNA. Okay, you'll see. Right here is a pre-mRNA, as you saw it before. 1 to 30 bases are here, 31 to 104 is here, and 105 to 146 is here. And then all of this come together by an enzymes called ribozymes. Intron from here to here, that gets split off, get cut off. This one get cut off. And these two, they're called introns. They stay inside of the nucleus. But these guys will come out. And then, of course, you have the poly-A tail at the end, and you have the 5' cap. Okay. Here is a spliceosome, that protein molecule that I was talking about right here. That's spliceosome. And then it goes through the mRNA. This is the mRNA right here. This is the mRNA. And then spliceosome will splice this segment right here, and they get cut off. They stay in the nucleus. But these segments right here, exon 1 and exon 2, they are combined together by ribozymes. Okay, the enzyme ribozymes. The functional and evolutionary importance of intron, you know, I'm going to let you guys read this by yourself, and then, you know, the introns had function at one time, and throughout the evolution, they do not have a function now anymore, so they do not get translated. Proteins, however, modular actually are consisting of the discrete regions called domains. In many cases, different exons codes for different domains in a protein. Exon shuffling may result in evolution of a new proteins as well. So anyhow, these are different domains and different things. So please read it. A cell translates as mRNA messenger into protein is okay. Now we are getting into transfer RNA. Transfer RNA look like a clover leaf. Okay, and then what it does, it grabs an amino acid in the cytoplasm and bring it to the ribosome and unload it and moves away. So this is the ribosome, my fist. This is a tRNA. It grabs the amino acids, brings it here, unload it, and goes away. Another tRNA comes in, unloaded, goes away. And these amino acids are going to be attached to each other by peptide bond, which you studied in the first exam. So tRNA transfer amino acids growing, T stands for tRNA, mRNA stands for messenger RNA, aids to the growing polypeptide in ribosomes. Molecules of tRNA are not identical. Each carries a specific amino acid on one end, and then each has an anti-codon. So codon was on mRNA, and tRNA right here, this is an mRNA. So this is a codon, right? Three of them. And the tRNA comes in, it matches. You will see some pictures here in a minute. These three are called anticodon, right here. So these are tRNAs, they have to match. Remember, if they are not matched, they do not unload their amino acid. They will match AAA to UUU and CCG to GGC, UGG to ACC, already unloaded and went away. This one is coming. So amino acids are being attached to each other: phenylalanine, tryptophan, glycine, serine, and here is a peptide bond between them. Peptide bond, peptide bond, peptide bond, peptide bond. I hope I'm making sense. And of course, this is ribosome, right? This area, that is your ribosome. Ribosome, mRNA, it goes like this. tRNA comes in, unload the amino acid, it moves, tRNA comes in, unload amino, another tRNA comes in, unload amino acid, it moves. Another tRNA comes in, unload amino acid, it moves. That's how it happens. That's what it's happening. Okay, tRNA molecules consist of a signal RNA strands that is only about 80 nucleotides long, flattened into one plane. He's talking about the clover leaf that I talked about it, the L shape and so on and so forth right here. This is a clover leaf model. This is the L shape model. And at the end are anticodon, 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 which has to match the codon. And still remember, there is no T in here also. It's U instead. So accurate translation requires two steps. First, a correct match between tRNA and amino acids done by synthesizing amino acid tRNA synthase. And then the second, a correct match between tRNA, anticodon, and mRNA, which we already talked about that. Flexible pairing at the third base of a codon is called wobble and allows some tRNA to bind to more than one codon right here. So this is the amino acid and tyrosine amino acid tyrosine, and this is tyrosyl tRNA synthase. And then what happens, as you can see, it comes in here. They put them together, this molecule, what this PowerPoint is saying, this molecule, put that tRNA and that amino acids together. And right here, they're showing you as the wobble, so on and so forth. Okay, ribosomal, the third RNA is ribosomal RNA, which there is not much information about it, but it seems like it's synthesized, of course, in nucleolus, and it goes into ribosome, right here, ribosomes, and just monitor the whole entire process of translation. Okay, so you can read up on it again. You should know something about it, and that's pretty much it. I'm not gonna say too much. So the three spots in ribosomes, EPA, E as for exit, P for the placement. I don't know, they didn't mention it in here. And A for amino acid, they are saying it A. P is for peptidyl, the placement of the amino acids they attach, look here, peptidyl tRNA binding sites right here, and A for arrival, amino acid tRNA binding sites. So what happens, the maximum you can have two RNA inside of the ribosome. One of them already left, the other one is arriving, and this one is placing the amino acids. And of course, your textbook is saying the reason is for P is peptidyl tRNA. I'm saying it easier for you, E as an exit and A as an arrival. That would be amino acid tRNA binding sites. Right here, they're showing it to you again, nice and beautiful. The mRNA is passing through, and tRNA brings in the amino acids right here, P site, A site, EPA. Okay, the P site holds the tRNA that carries the growing polypeptide chain. A site holds the tRNA that carries the next amino acids, and E is the exit. Building of a polypeptide, there are three steps: initiation, elongation, termination. So all three stages require protein factors that aid in the translation process. Energy is required for some of the steps, like here, GTP becomes GDP, that's equivalent to 1 ATP. We already talked about it at the beginning of semester. So right here, they didn't show in this diagram, they didn't show you the large ribosomal subunits, but it is there, large ribosomal subunits there. Initiation, methionine, remember that? It has to be AUG. Okay, so that's pretty much is the initiation, translation initiation. And then after that, the elongation is codon recognition, translocation, and energy expenditure occurs first and third steps. And then, of course, translation proceeds along the mRNA in a right here. They're showing it to you nice and beautifully. Nice diagrams. That is how things are happening in here. And then you need a GTP at the beginning right here. But when the amino acids are attached, then they're exiting, you need another GTP. Termination occurs when a stop codon in the mRNA reaches the A ribosome and so on and so forth. Again, I'm not going to read the rest of it for you guys. I think I feel like I talked about all of these. And at the end, of course, there is a protein molecules which break down the ribosome, split them into the parts. Often translation is not sufficient to make a functional proteins. That's right. It goes to Golgi apparatus and get remodeled and so on and so forth. Right here, they're showing you either in the rough endoplasmic reticulum or ribosomes. So signal recognition particles, they're right here. And they say the protein molecules needs to be folded and carbohydrates added, so on and so forth. There is a lot that can be done either in rough endoplasmic reticulum or Golgi apparatus. All of those modifications happens in those. Multiple ribosomes can translate a single mRNA, simultaneously forming a polyribosomes or polysomes. Polyribosomes or polysomes enable a cell to make many copies of polypeptide very quickly, and they are showing it in this transmission electron micrograph. Again, they're showing that. And right here, what I talked about at the beginning of the lecture, right here, they are putting. We didn't talk much about gene regulation, but pretty much the central dogma in some detail. The central dogma of biology is right here. They're showing it to you guys. Let's watch this. Huh?
This student is cramming for her biology test with only a frosting-covered donut for lunch. Specialized cells in her pancreas respond to the increasing amount of sugar in her blood by releasing insulin, a small protein that regulates blood sugar levels. Let's go inside one of these cells to see how this protein is manufactured. The instructions for making insulin are coded by a segment of DNA in the nucleus. In transcription, an enzyme zips along the DNA, forming RNA, shown here in red. RNA nucleotides line up with their complementary DNA partners, transcribing the information in DNA into RNA. As the RNA grows, it is processed in several ways. First, a modified guanine nucleotide is added to the beginning as a cap. Also, segments of the RNA strand that do not actually code for the protein are removed, and the remaining segments of RNA are reconnected. Finally, extra adenine nucleotides are added to the end of the RNA strand, forming a tail. The completed messenger RNA, mRNA, now leaves the nucleus. The message in mRNA is translated into a protein in the cytoplasm. First, a transfer RNA, tRNA, arrives, carrying a specific amino acid. The small subunit of a ribosome attaches to the mRNA. Now, the large subunit of the ribosome attaches. A second tRNA docks, bringing another amino acid. The ribosome helps to form a covalent bond between the two amino acids. The mRNA shifts, and the first tRNA leaves. A new tRNA brings another amino acid. The ribosome helps to form a new bond, and the process is repeated. Notice that one end of a tRNA molecule has a set of 3 bases called an anticodon that pairs with complementary bases on the mRNA. The other end of the tRNA carries a specific amino acid. Different types of tRNAs carry different amino acids. In this way, the message in mRNA is translated into a specific sequence of amino acids. For proteins that will be secreted from the cell, like insulin, the ribosome docks on the rough ER, and the protein grows into the ER compartment. The new protein molecules are packaged in a vesicle that is transported to the Golgi apparatus, where many proteins are processed. However, insulin is packaged in a vesicle that leaves the Golgi and is then processed. Proteins secreted from the cell are shipped to the plasma membrane. Here, insulin is secreted from the pancreas and begins to regulate the student's rising blood sugar levels.
You know, they didn't mention in this video that even insulin we have pre-insulin, pro-insulin, and then we have pre-insulin. Even insulin will be modified later on. But anyhow, they didn't talk about it. So here, here's another video. Okay.
In translation, a cell reads an mRNA message and assembles a polypeptide accordingly. We can divide translation, the process of building a polypeptide, into 3 stages: initiation, elongation, and termination. Translation is initiated when the small ribosomal subunit binds to the leader at the 5' end of the mRNA molecule. The anticodon of the initiator tRNA binds to the start codon, AUG. The initiator tRNA always bears the amino acid methionine. Proteins called initiation factors help bring the mRNA, the initiator tRNA, and the small ribosomal subunit together with the large ribosomal subunit to form an initiation complex. The initiator tRNA sits on the P site on the ribosome. Elongation of the polypeptide now occurs, with amino acids being added one by one. The codon in the A site of the ribosome pairs with the anticodon of the appropriate tRNA molecule. Part of the ribosome catalyzes the formation of a peptide bond between the amino acid extending from the P site and the amino acid extending from the A site. The tRNA in the A site now translocates to the P site. The tRNA that was in the P site moves to the E site and exits from the ribosome. Meanwhile, a new tRNA brings its amino acid to the A site and the process is repeated.
As I showed it to you guys like this.
3 special base triplets, UAA, UAG, and UGA, do not code for amino acids, but instead act as stop codons, which terminate the process of translation. A protein called a release factor binds to the stop codon in the A site. It acts to free the completed polypeptide from the tRNA that is in the P site, and the translation assembly comes apart. The polypeptide becomes folded, modified, or combined with other polypeptides to form a functional protein.
Okay, the only thing I did not mention so far was that protein molecule. Now it's slipping my thoughts. Mutation, anyhow, that yellow protein molecule is called polypeptide protein molecules. It comes and sits on the A side. So the whole 9-yard knows this is time to quit, to stop, and that's the end of it, okay? So mutation, as we already talked about it, if there's only one amino acid is different, the protein molecule can be completely different. So there are different types of mutation. He's talking about it. It's point mutation and frame shift, and you will see all of those. So for example, in the case of sickle cell, it is a point mutation. It's a point mutation. the DNA is CTA, and right here that changes to A and T, and of course the mRNA is going to be GUA. This is the normal one, GAG, and then GUA, it gives you valine instead of glutamine. So if it is glutamine, then glutamic acid, then it's normal, and if it is valine, it's abnormal. It's look like a sickle cell just because of one amino acid difference and one nitrogenous base difference. Okay, anyhow. So point mutation within a gene can be divided into two general categories: nucleotide pair substitution, one or more nucleotide pair insertion or deletion can occur. Here there are a few different type of substitution: nucleotide pair substitution, silent mutation. If you have UCU and UCG, they both give you serine. Imagine the first one is supposed to be the normal one. Okay, but if a mutation occur and instead of UCU it's UCG, it still gives you serine. Okay, missense mutation still codes for amino acids, but not the correct amino acid, does not affect the protein that much. So in this case, in case of sickle cell, you saw valine, it makes the protein, the shape of the cell and everything drastically different. But sometimes instead of, you have glutamic acid, valine doesn't make that much of a difference. It's a missense mutation. Nonsense mutation, that is when you have leading to a non-functional protein, which is changing amino acids codons, a stop codon nearly always, and that is a nonsense. The stop codon comes instead of the amino acid, and that's again a horrifying mutation. Then wild type here and nucleotides. Again, we're talking about different type of mutation, insertion and deletion. If you insert or you delete, we talked about these a little bit for the chromosomes. You remember that? But here are on a DNA level. Okay, and then you have frame shift, which mutagens, any chemicals or any physical things that cause the sequence of DNA changes. The idea of a gene evolved through the history of genetics. We have considered gene as a discrete unit of inheritance, a region of specific nucleotide sequence in chromosomes, a DNA sequence that codes for a specific polypeptide gene. A gene can be defined as a region of DNA, which I gave you, can be expressed to produce a final functional product that is either a polypeptide or an RNA molecule. Here they are. That's what I was waiting for. This is the polypeptide, and these are for examples of a silent, silent and missense, and then what else? What else do we have? Nonsense and frame shift. Okay, study these things a little bit. Another example of frame shift, and then 3 nucleotides, pair codons, missing. So he's talking about all of these things, and then is this video? No, I don't think so. That's the end of the chapter, you guys. Study it, please know it, and then if there's any question, holler at me.